Thanks again to everyone who's taken time to join us this morning. So today I'm going to be talking about some research that I've been doing here up in Virginia on Lyme disease ecology in conjunction with some of my students here at Hollins, working to understand this newly emerging hotspot that we're seeing here in southwestern Virginia. So some basics on Lyme disease, and I'm sure many of you are familiar with. There's about 30,000 cases that are reported to the CDC on an annual basis, but it's estimated that there's likely closer to 300,000 cases occurring here in the US. And as you can see in the graph on the top right there, the case incidence has increased dramatically over the course of time. And the causative agent of Lyme disease is Borrelia burgdorferi, with the vector being actually a number of different Ixodes tick species. But the species of most human relevance here in the eastern United States, of course, is Ixodes scapularis, or the black-legged tick, which many people commonly call the deer tick, which many tick biologists cringe at because black-legged ticks tend to feed on many other things besides deer. So black-legged tick is the common name that we use for Ixodes scapularis. And one of the things that's really fascinating from a disease ecology perspective about Lyme disease is its distribution versus the vector distribution. So we can see on the top right map here, these are cases of Lyme disease that have occurred here in the eastern portion of the US. And you can see that, of course, Lyme cases tend to concentrate in northeastern United States and the upper Midwest, with very few cases in the southeast. But what's interesting is if you look at the map here on the bottom right-hand side of black-legged tick distribution, you can see that they are actually distributed throughout much of the eastern United States. So one of the really big questions that has emerged is, why is that, right? Why don't we have Lyme disease in southeastern United States when the tick vector is there? There's been tons of research happening in that regard for a number of years. And there's two main hypotheses for why we see this discrepancy. The first one is that it is suspected that black-legged ticks tend to use more competent reservoir hosts in the northeastern United States. So black-legged ticks, particularly larvae and nymphs, tend to feed primarily on white-footed mice in the northeastern US, which are wonderful reservoirs for Borrelia burgdorferi, which causes Lyme disease. That's in contrast to southeastern United States, where it's suspected that black-legged ticks are using other wildlife hosts, such as skinks and lizards, which are not great reservoirs for Borrelia burgdorferi. So that's one thing. The other piece is there's been a lot of research done in the last 5 to 10 years that has been examining the behavior of black-legged ticks. And something that we know about black-legged ticks is that there's actually a distinct northern and southern population of black-legged ticks. And beyond that, there seems to be distinctive questing behaviors, where questing behavior is basically what ticks do to get on a host. So they tend to crawl up to the tips of vegetation, waiting for a wildlife host or a human to brush up against them so that they can then get on and bite that host. So what's been recognized is that black-legged tick nymphs in the northeastern United States tend to quest more aggressively than black-legged tick nymphs in the southeastern United States. So what that means is, if they're questing more aggressively, that means that they're questing higher up on vegetation and perhaps questing more frequently, which is therefore going to make them more likely to be found on a human, right? They're more likely to be able to get on a human, whereas black-legged tick nymphs in the southeastern United States tend to stay down low in the leaf litter, where they're unlikely to encounter humans. And beyond that, the other piece is, I've mentioned nymphs, and it really is the nymphs that tend to have this unique questing behavior difference. Nymphs also are the life stage of black-legged ticks that are most associated with transmission of Borrelia burgdorferi Lyme disease to humans. And that's because they're the first life stage that's capable of carrying Borrelia burgdorferi. Larvae do not carry the pathogen, and they're small. So it's really easy for a person to not see them, and therefore the tick is less likely to be removed and therefore successfully transmit the pathogen to humans. So really important that we're seeing these behavioral differences as well in the nymphs. Something else that's sort of interesting from an ecology perspective is the expansion of Lyme disease that we've seen over time. So over the past 20 years or more, we've seen Lyme disease spread throughout the northeastern US, westwards towards the upper Midwest, but it's also began to move a little further southwards into Virginia. So you can see in 2001 here that there were very few cases here in Virginia. But by 2017, we're beginning to see more and more cases, particularly along the western portion of the state, with this unusual hotspot forming here in southwestern Virginia. And so if we begin to dive in a little bit more to what's happening here in Virginia, interestingly, sort of going back and looking at cases, Brinkerhoff et al. Did this, they found that up until 2006, cases were relatively low in the state, but starting in 2007, which you can see here, we began to have a sharp increase in the number of cases. The other thing that was interesting was at that time, we began to see cases shift from the northern and eastern portion of the state over to the western portion of the state. And you can see that since then, the average annual increase along the western portion of the state has increased pretty dramatically, again with this hot spot here in southwestern Virginia. And what's especially interesting about this is that if you're unfamiliar with the geography of our state, the Appalachian Mountains tend to follow this western border here. So cases seem to be clustering along these high elevation areas in the Appalachians. And some research was done to try to better understand why this might be occurring. And so Brinkerhoff et al. Actually did a study on an east to west gradient across the state examining Ixodiscapularis nymphs. Again, they focused on nymphs because they are most significant for human transmission for Lyme disease. And when they did this work, these are the sites that they had. They had 4 sites located across the state. And what they ultimately found was that there were higher densities of Ixodiscapularis at this high elevation site labeled LE, and that there was also a higher prevalence of Borrelia burgdorferi in those ticks. So this was really interesting. It seemed to fall in line with the trends that we're seeing with Lyme cases tending to concentrate in these high elevation western portions of the state. But really, these conclusions were drawn based off of just that one site. So there needed to be more research looking at Lyme disease ecology in Western Virginia. And in particular, there really has been minimal to no research done here in Southwestern Virginia where that hotspot has developed. Now, when we're trying to figure out why we have this hotspot developing, and this would be true of anywhere where we're seeing an increase in case incidence of a disease, right? There's a lot of reasons that could be occurring. Could be occurring because of over-reporting. But other reasons that seem perhaps a little more likely is that it's possible that we have an increase or a greater abundance of the vector Ixodiscapularis. It's also possible that we have the specific populations of Ixodiscapularis that have been shown to quest more aggressively, e.g. sort of what we tend to see more frequently in the northeastern US. It's also possible that these ticks are carrying Borrelia burgdorferi at a higher prevalence. And then the last idea is that it's also possible that ticks are coming into contact with humans more frequently. Now, part of that could be because of the more aggressive questing behavior, but there could be other reasons that humans are coming into contact with ticks more frequently. For example, if we simply have individuals who are spending more time outdoors and or in the woods, which may be something that's playing a role here in the Roanoke Valley that I'll talk a little bit more about later. And the other option, which is probably the more likely option, is that there could be a number of things happening here in this region that are leading to the high case incidence that we're seeing here in southwestern Virginia. So I moved up here about 3.5 years ago, and when I first started sort of exploring all of this literature and seeing these trends, I thought, okay, let's sort of start ticking off these boxes and try to determine what is driving this trend that we're seeing. And really the first place to start was to get some basic baseline understanding of what the species composition and phenology was like here in the Roanoke Valley, because as surprising as it might seem, there actually really has been no work done on tick ecology here in this region. So the first step for me was to begin to understand what that tick species abundance and phenology is, and then to determine if there are any associations between species composition, in particular black-legged ticks, and elevation. So to do this, we established 12 different sites throughout the Roanoke Valley region, and in doing that, we then began collecting ticks on a monthly basis for a full calendar year. And of course, we also collected elevation at each site, and we attempted to have roughly half sites were above 500 meters and half of our sites were below 500 meters. There has been some work done previously looking at sort of what the maximum elevation might be that black-legged ticks can occur in. And that work, which was done based off of extensive field sampling throughout the eastern United States, had speculated that it seemed that black-legged ticks did not occur above roughly 500 meters. But with the Brinkerhoff et al. Work that was done on that east-west gradient across the state of Virginia, they speculated that ticks, black-legged ticks, seem to be most common at higher elevations, but importantly, their particular sites were just under 500 meters. So given that we were seeing, the clustering of cases along the Appalachian Mountains here in Virginia, and there are many locations within the Appalachian Mountains here that are above 500 meters, we were curious to see if there were ticks occurring above that sort of threshold that had been reported in the literature previously. So we collected those ticks on a monthly basis. We identified all of them for our larvae and our black-legged tick nymphs. We confirmed their ID with 16S PCR and sequencing. From there, we were able to sort of figure out what was going on. So in total, we collected around 20,000 ticks, right? And we weren't just interested in black-legged ticks. We wanted to get a full picture of what was going on with the general tick ecology in the region. But for the purposes of our talk today, I'm really focusing in on black-legged ticks and Lyme ecology. And you can see black-legged ticks were common. They're second most common species that we collected, second to Amblyom americanum, which is not surprising for this region. And you can see we were able to capture about 134 adults, 306 nymphs, and just over 1,500 larvae. And I will say we're about done with all the 16S sequencing for the larvae. So most of those are indeed Exodiscapularis. So when we began to look at phenology for our tick species, right, again, we're most interested in this particular talk anyways, in our Exodiscapularis, right, which is abbreviated IS here in the legend. And so you can see that our Exodiscapularis adults tend to be active starting in November through December. They dip off a little bit in January, and then continue on through February. And that's really the type of activity that we expect to see. And it's important to highlight that, right? Because a lot of times folks think, oh, it's wintertime, it's cold, ticks aren't out. Well, yes, they are. And in fact, black-legged tick adults are out. So Lyme disease risk is still present. But the other thing that was really fascinating to us and was sort of what we were expecting to see, but it was nice to actually have validation, was that we are seeing a very distinct, pretty high peak in black-legged tick nymph activity, right? And that was exciting because of those behavioral differences that have been observed in northern versus southern black-legged tick nymphs. So, for example, I did my graduate work down in southwestern Georgia using the exact same methods that I used on the current project to collect ticks on a monthly basis down there. And what was interesting to me was that when you look at the abundance of adults that we collected in southwestern Virginia versus southwestern Georgia, you can see that the abundance is kind of similar, right? Probably not significantly different, right? But when we look at nymphs, we see that I collected 0 nymphs in southwestern Georgia. And I will say that that is out of close to 50,000 ticks that I collected for that project. In the meantime, in southwestern Virginia, I collected roughly 12 ticks per hour, right, when we're talking about the nymphs. So this seems to indicate that we are seeing those sort of northern behaviors that are associated with black ticks in the northeastern United States with that aggressive questing behavior. And I will say we're working on actually comparing our results to some phenology data that's occurred in the eastern portion of the state. And I can anecdotally tell you that their black-legged tick data tends to look more like the data we're seeing here in Georgia. They just don't get black-legged tick nymphs. So it seems to indicate that we have both populations here in the state of both the aggressive questing ticks here in the western portion of the state and the less aggressive questing ticks in the eastern portion of the state. And that's probably playing a very big role in the differences in Lyme incidence that we're seeing across the state. Now, we also looked at elevation with our black-legged ticks, and we found no significant difference in abundance above and below 500 meters with black-legged ticks. So that does seem to refute the idea that black-legged ticks cannot occur above 500 meters. But it also doesn't necessarily indicate that they prefer high elevation either. I'll talk a little bit about some of the limitations in our particular study with that conclusion, but again, no significant difference in elevations that we were able to detect. So again, I mentioned, right, this lack of correlation with Ixodes scapularis abundance and elevation. They do occur above that magical, it was specifically 510 meters was the cutoff that's been reported in the literature previously. But I will say that our data is limited, right? At the end of the day, we are in the Roanoke Valley, which tends to just be at a higher elevation than if you were to, say, go to the eastern portion of the state. So our specific sites ranged between around 300 meters and 570 meters. So it's possible if we were able to include even lower elevations by moving even further eastward, maybe we would begin to see some correlations or differences in abundance, with elevation. But based off of the work that we did anyway, we weren't really observing that. The other piece that was really fascinating for us is that, yes, Ixodes scapularis nymphs do seem to be exhibiting behavioral traits that are more closely associated with the ticks that we see further in the northeastern United States. And that does not seem to correlate with the behavior that other tick ecologists have observed further east and into the state. And that seems to correlate with sort of where we're seeing Lyme cases as well. So to answer sort of this big question of, what seems to be driving the emergence of Lyme disease here in southwestern Virginia, it certainly seems like part of it is this more aggressive questing behavior of our Exodia nymphs. The other piece is Roanoke is a city, albeit a small one. We have a fairly large population here, particularly compared to other regions of Western Virginia. And importantly, it's oftentimes assumed, right, that urban areas, you're going to be at lower risk for ticks because A, there's just less wilderness, B, people aren't spending as much time outside, etc. But that's not exactly the case here in Roanoke. Roanoke markets itself on an outdoor recreational lifestyle. People move here to be outside. We've got tons of national parks, state parks. We've got some of the best mountain biking in the country, lakes, Appalachian Trail. We've got it all. And so we have a large population, many of whom are seeking outdoor recreational activities. And because of that, I suspect that we also simply have people that are coming into contact with ticks more frequently because of that. So I think all these things are playing a role in the higher Lyme incidence that we're observing. There are still questions to ask. There are still other things that could be contributing to this in addition to the observations that we've made to date. One of the big questions we of course have is pathogen prevalence, right? What's the prevalence of Borrelia burgdorferi here? So the work is still very much ongoing. We're actually very close to wrapping up pathogen testing of our ticks, testing for Borrelia. And so we'll be excited to have those results soon. The other piece that we're working on right now in our lab is doing a basic phylogenetic analysis of the Exodia scapularis ticks that we've seen so that we can compare them to ticks that have been collected elsewhere in the state in hopes that we can maybe gain a better understanding of sort of how our ticks might be related to ticks elsewhere in the state, sort of movement, origins of these ticks, etc. And then also some behavioral studies to sort of confirm what seems to be occurring here in terms of having these ticks here in the western portion of the state exhibiting this more aggressive questing behavior as compared to ticks in the eastern portion of the state. We've done some initial work with that. And are hoping to continue with that work in the next year or two. So kind of shifting gears now, I think one of the big questions that I'm sure many of our listeners today have is what role can land managers play in reducing risk of Lyme disease and just reducing risk of tick-borne diseases in general, right? And there's been gobs of research done on potential tick control methods that are out there, right? You've got burning, herbicide, acaricide, mechanical clearing, right? All kinds of different approaches that have been investigated for their efficacy in reducing risk of tick-borne disease. There was a really great review that came out back in 2018 by White and Gaff that actually evaluated all of these different tick control studies to see sort of what their efficacy was. This particular paper focused on the three most common human biting ticks in the eastern US. So they focused on blacklegged ticks, Lone Star ticks, Amblyomma americanum, and then the American dog tick, Dermacentor variabilis. So there's tons of information in there, and I highly recommend you read it if that's something you're interested in. I tried to kind of summarize the gobs of information that was in that review for you all today. So focused in on the data on black-legged ticks, and in particular looked at studies that saw at least a 50% reduction in some life stage of black-legged ticks, and that reduction was sustained for at least a year, if not longer. So that was just sort of the criteria that I used. And you can see, right, your eye might immediately jump to, right, where we saw the highest efficacy, which was bait boxes and tick tubes, right? And I guess one point of clarification: these are these are all studies, right? So when I say 4 out of 4, that is 4 studies found that reduction, 50% reduction for over a year out of the 4 studies that they examined that were looking at bait boxes. So right, this looks great. Wow, 100% of the studies, 4 out of 4 studies found bait boxes work. Now, first off, you might be asking, what are bait boxes and what are tick tubes? So just to go back to this previous slide, so this picture here is of a bait box, and both of these methods are targeting small mammals, which again are, particularly in the Northeast, the primary reservoirs for Lyme disease. These are the hosts for our immature ticks, so our larvae and our nymphal ticks. And so the idea with the bait boxes here is that you have some bait in a box, aptly named, and importantly, as the mouse comes in, it has to go sort of under this tunnel, if you will, that is coated in an acaricide. So basically, the mouse is wiped down with an acaricide that will kill any ticks on it, and voila, you have freed that particular rodent of ticks. Tick tubes have a similar premise. There's a lot of do-it-yourself instructions for how to make your own tick tubes online. There are also some commercially available tick tubes as well. But again, aptly named, they are tubes, and there's acaricide-treated cotton inside of them that the mouse would go in, use this cotton for nesting, and in the process get coated again in the acaricide that the cotton's treated with, removing all the ticks from the mice. So that sounds really cool, right? But as you might imagine, the efficacy of these on a landscape level is, minimal, right? So the idea that one would be able to treat really large swaths of land with bait boxes and tick tubes, right, and to do it on a consistent basis is low, right? Because you're going to have to constantly be refilling the bait in the bait boxes. You're going to have to constantly be refilling the acaricide in both the tick tubes and the bait boxes, and of course the cotton with the tick tubes. And you have to have enough of these out that you're treating a significant proportion of the mouse rodent population. So these seem to be really effective and promising when we're talking about using them, say, in a residential setting, right, for someone's yard or something of that nature, but maybe not necessarily on a large landscape level. There's also some concern with the bait boxes in particular, that you are providing supplemental food to the primary reservoir for Lyme disease, which is probably going to lead to increases in mouse populations, right? Even with the tick tubes, you're providing additional refuge for mice. All of this can lead to increased survival, increased population numbers, and that's a little bit concerning as well. So you might say, okay, well, let's move on to the next group that looks promising. And these are all methods that are focused on deer, right, which are really important hosts, particularly for adult black-legged ticks. Deer removal implies culling or increased hunting pressure on deer. Deer fencing is fencing deer, out of an area. And then the 4-poster method is, again, I'm going to go back to this other slide, is what you see here, where you basically have bait stations. There's bait here, and if you were to look over here, if you could see that far, there's another bait station right here. And you've got these posters that, again, are soaked in acaricide so that when the deer come in, they brush up against these acaricide, rollers or posters. And again, it removes ticks from the neck and the head, which is where you see ticks primarily attaching on deer. So right? When we look at that, again, seems pretty promising. But again, there are some concerns and some logistical constraints there, right? So with deer removal, you typically have to remove a very large percentage of the population to have any success. And as you might imagine, there may be some public outcry about that. You then have fencing deer out, right? But right, fencing deer out of a very large piece of land is probably not logistically feasible. And there's also, ecological concerns about removing one of the primary herbivores from an ecosystem. So that leaves the 4 poster methods. But again, the logistical constraints there are pretty large. You're constantly having to refill bait as well as to reapply the acaricides. You have to have enough of those stations that you are treating almost 100% of the population to see efficacy. And so all of those things mean, right, if you were to sustain that on a long-term basis, it's probably going to be pretty challenging. The other concern with both the 4 poster method as well as the bait box and tick tubes is this concern about developing resistance to acaricide. So could ticks develop acaricide resistance? That obviously would be a very big issue. So the last one we're really left with that seems to be effective is prescribed burning, where we saw 25% of the studies had some success with this on a long-term basis. And so I'm going to focus in a little bit more on that. And I gave a webinar on the research that I did examining prescribed fires and ticks a number of months ago with NAFC. So if you're interested in hearing more about this particular research that I did, I encourage you to find that webinar where I go into much more detail, or I'm happy to answer questions about it if anyone has questions today. But I thought I would just real quickly talk about it. So there's been lots of studies done on the impacts of prescribed fire on ticks, not just the 4 that you saw from that review. There's dozens of studies out there. And all of those studies tend to agree that yes, tick populations are reduced immediately after fire. So fire does kill ticks. But all studies have found that the tick populations tend to steadily recover over time to some degree. But where the studies tend to begin to kind of sort of diverge is what the long-term effects of fire are. So ultimately, right, a year, 2, 3 down the line, some studies were seeing decreased tick populations. Others found that the tick populations just rebounded back to their pre-burn levels. And yet others actually found increases in tick populations. So when we began thinking about doing a project on ticks and fire, of course, one of our immediate questions was why the discrepancy in the literature? Why are we seeing so many different outcomes with these previous studies? And what we found is that most of the previous studies tended to either not account for other variables that we know affect tick populations like wildlife host abundance, vegetation structure, microclimate, etc., and or they did not simulate real-world management practices. In particular, many of the studies were done on very small scales, so really small areas were burned, say like an acre or less. Other studies, they just did a singular burn in an area that perhaps had never been burned before, and that was it, right? And that's really different from what we see in management scenarios where you're burning typically dozens, if not hundreds or thousands of acres, and you're doing so on a regular basis, right? Anywhere from every 2 to 5 years, if not more. And that has distinct implications for the ability of a tick population to be able to recover, right? Whether you're burning a small patch of land versus a big patch of land, whether you're doing repeated burns or a single burn, right? So what we did for this particular project was we established 21 different sites in southwestern Georgia. Some of those sites had been subjected to long-term prescribed burning, meaning that they had been burned for at least 10 years, with burns happening everywhere from every 2 to 5 years there, every 2 to 4 years. Or they were unburned. And if I say unburned, I mean that they were unburned for a minimum of 10 years. And we went in and we collected ticks in those different sites for 2 years straight on a monthly basis. We ended up collecting just shy of 50,000 ticks. And we also tested them for pathogens. And what we found was that, yes, areas that burned had significantly lower numbers of ticks that were infected with pathogenic bacteria, meaning bacteria that can cause disease in humans, versus our control sites, which is abbreviated UBUB here. Right, so you can see at our burn sites, like in 2011, there were no ticks that had pathogenic bacteria that we collected, and in 2010, there were very few, right? So even at the peak, we're talking 0.1 ticks per hour that we encountered that were infected with pathogenic bacteria. And that's in direct contrast here where we have a very different scale, right? With our control sites, where at peak tick activity, we're seeing approximately an encounter rate of 1 tick per hour with pathogenic bacteria, right? So really significant differences in disease risk at our unburned sites versus our burned sites. So in terms of the conclusions that we were able to draw there, right, ultimately we found that long-term prescribed fire does reduce tick populations regardless of burn interval, host abundance, and vegetation structure, which we did account for in that study. And ultimately what we observed was a 98% reduction in encounter rates with ticks infected with pathogenic bacteria. And one thing I want to really emphasize is we saw sustained reductions, right? So if you look back here, right, ticks just simply weren't in these sites. So it wasn't that they were getting knocked back and then they returned. They just weren't there for the whole 2 years that we were there. I mean, they were there, but right, in very low numbers. So that was interesting to us because that hadn't really been seen in the literature previously. And so our big question was why? There seems to be something beyond just the fires that is suppressing the tick populations at these sites. And I will say that the reduction in these encounter rates was primarily because of reduction in tick numbers, not necessarily because of changes in pathogen dynamics. But there were some exceptions to that, which I'm happy to talk about if anyone's interested. But we ultimately did an experimental field study to try to better understand why we were seeing these sustained reductions in tick populations in the burn sites. And what we ultimately found was the change in vegetation structure that was created by the use of long-term prescribed fire was what was making these sites unhospitable to ticks. So if you think about fire, right, you're reducing the understory, suppressing the midstory, in our case, allowing a beautiful semi-open pine canopy. And this allows more sunlight to penetrate through the canopy to the understory, which creates a hotter, drier environment. And what kills ticks? Desiccation, drying out kills ticks. And so it just turns out that ticks could not survive in those burned habitats because of that hotter, drier environment, right? And what was important was this really emphasized the need for long-term prescribed burning, right? You cannot achieve that type of forest and that type of vegetation structure without fire, and you can't maintain that type of forest structure without fire. So really underscored the need for consistent long-term burning, not singular burns. And in fact, we suspect in some cases singular burns could actually be detrimental in terms of reducing or not reducing, perhaps increasing risk of tick-borne disease. So this was really exciting and definitely has potential for reducing tick-borne disease risk in other regions and ecosystems, but that work has to be done, right? So there's still a big question about would we see these types of reductions happening in other ecosystems in other regions of the country with different climate? And in particular, could this be a tool used in Lyme endemic areas to reduce risk of Lyme disease? A lot of research still to be done, and I very much hope to be able to do some of that here now that I'm in Virginia. So stay tuned, hopefully, for some info on that. The last thing I feel I would be remiss without talking about, and then I want to be sure to leave time for questions, is for my many land managers and field biologists who are out there. A lot of this will be old news to you, but just how to keep yourself safe, how to reduce your own risk of tick-borne diseases, right? So the first thing is you got to keep ticks off of you, right? So using tick repellents, DEET, permethrin, whatever you might have. Right, the permethrin's nice because you can treat your clothes and it lasts typically for a couple weeks or so many washes. The other thing I will mention that actually Amanda turned me on to that I was not aware of until we were having this conversation last week was for those of you who do prescribe burns, importantly, DEET is flammable and permethrin is not. So if you're working with fire, permethrin may be your tick repellent of choice. Right, wear pants, closed-toed shoes, and long sleeves even in the summer, right? Because that's when the ticks are really out. And if you want to look really cool, you can tuck your pants into your socks. Yes, I really do that. And while you're out, in the field, check your clothes occasionally. Look down at your pants and see if you see anything crawling on you. So that's your first line of defense, right? Once you get home or, back to the office, if you're able to, promptly change your clothes and take a shower and do a full body tick check, because despite our best efforts, there will always be ticks that manage to get through those different defenses and bite you. And if there was only one thing that I could do to try to protect myself, it would be to do a full body tick check as promptly as I can after returning from working outside. So if you find a tick on you, remove it as soon as you can. If you've got kids and you're going out with them, outside of work, don't forget to check them as well. And if you find a tick, right, do's and don'ts of removing ticks, always, if you can, use fine tip forceps. Grab the tick as close to the skin as possible. Avoid squeezing the body. That can cause them to regurgitate pathogens into you. When you remove the tick, just a slow and steady straight pull out, no twisting, no jerking, which could result in accidentally leaving the head behind. And all of those wives' tales you hear about, burning ticks off, drowning them in alcohol, Vaseline, any of that stuff does not work and may increase the likelihood of the tick transmitting any pathogens that it might have. So definitely don't do those things. Once you've removed the tick, clean it like you would any other sort of wound, and then keep the tick and look at it. Inspect the level of engorgement, right? So I've got a handy little photo here of sort of the different levels of engorgement of a tick over time. If the tick is not engorged, you're not necessarily out of the woods, but if it is engorged, that's definitely not a good sign, because that means it's probably been attached to you for at least 12 to 24 hours, and most tick-borne pathogens can be transmitted after 24 hours. Again, regardless of engorgement level, there are some tick-borne pathogens that can be transmitted in less than 24 hours. So again, you're not completely out of the woods if it's not engorged. Keep all ticks that you have found attached to you for at least a month, because that's how long it might take for symptoms to develop. And if you do happen to get sick, certainly seek treatment from your doctor immediately. And you can also have your tick identified and or sent out for testing for pathogens. So definitely value in keeping the tick. In terms of how to keep it, you can preserve it in alcohol or ethanol. You can also freeze it. So if you want to stick it in a Ziploc bag and freeze it, you can do that as well. Be sure to write the date and who the tick came off of. And with that, I think that's everything I have for you all today. I'd be remiss without thanking the many folks that made the tick work here in Virginia possible. In particular, I want to highlight my student Sierra Morris, who this was really her project, and she did a tremendous amount of work on it. And of course, the many partners that we've had that have allowed us to do work on their land and fund the work. So many thanks. And for the work out of Georgia with the prescribed fire, again, my collaborators, Michael Yabsley, Roy Burgess, Mike Connor, Michael Levin, and Galina Simpsova, as well as, again, our many partners in funding and use of land. So many thanks. With that, I'll leave you with some fun photos from around the field, and happy to take any questions.
Wow, thank you, Liz. That was really fascinating, and thank you for that great overview and all the different topics within this that you've touched on. We did have several questions come in. I think I'll just go in order.
Sure.
And we'll see what we can cover. So first, Joel asked, could this spread of ticks because, and this is back when you're kind of covering the study in the Roanoke area, could the spread of ticks be caused by global warming, which is possibly leading to more movement activity?
Yeah, so there's a number of reasons that we suspect we're seeing an increase in tick-borne disease cases, across the board, not just with Lyme disease. And one of those certainly it's been speculated is climate change, particularly in the Northeast and on up into Canada. We're certainly seeing both Amblyomma americanum moving further northward and Exodia scapularis populations moving further northward as well. Right, as temperatures become warmer, they're able to sort of migrate further northward. So we are seeing those expansions in tick species ranges. There's other things that we think are contributing to increases in cases as well. For example, changes in land management, changes in wildlife host populations. But yes, climate change is also certainly something that's very much a part of the conversation as well.
Got it, thank you. The next question that came in was about your collection method. This might be appropriate to one of the photos in your last slide there. What was your collection method? Are there lures or special equipment?
The good news is there's not a lot of special equipment. It is really low tech. So yeah, the photo here you can see is what I use. I use flagging. Most tick folks either drag for ticks, which is basically this piece of material, literally you drag it behind you and transects, or you can essentially do the same thing with a flag, where you basically are sweeping the vegetation and sort of, again, either walking in transects or working within a confined space and a defined space and then standardizing by time. So those are the methods I use, again, is the flagging, and I found that's more effective in a broader variety of habitat types. And there's other ways you can collect ticks is with dry ice or CO2 traps. But generally, the literature for the most part will advocate for dragging and flagging being most effective, depending on the species and the life stages that you're interested in.
Got it, thanks. Another question was, have you collaborated with veterinarians at all for collection? Emily, who's in the Asheville, North Carolina area, says, I know of one vet in my region assisting in tick research.
Yes. I haven't personally worked with veterinarians in my own research, but yes, I am very much aware of a number of different studies and research projects where veterinarians, both wildlife and domestic animal veterinarians, have certainly been able to contribute to tick collections and or testing animals for tick-borne diseases and things of that nature. Yeah, there's a lot of really great work that's been done with veterinarians for sure.
Got it, thanks. We had a question from Tom earlier. So does this mean that there is something in the ground and or the vegetation that the ticks are picking up to cause them to be carriers of Lyme disease?
Oh, good question. Basically, the way that ticks get Lyme disease is, I mentioned before, there's 3 life stages to for ticks, at least for human biting ticks here in the eastern U.S. Right, so you have eggs. It's a clutch, e.g., like a group of eggs, typically hundreds, if not thousands of them. They hatch into larvae. Then they turn into nymphs, and they turn into adults with a single blood meal on a host in between each of those different life stages. So when larvae hatch, they do not have the pathogen, but larvae have to have that blood meal on a wildlife host. And when they go and feed on a host, if that host is infected with Borrelia burgdorferi, the pathogen, the tick then becomes infected. It'll fall off the host after it's fed, molt, turn into a nymph, and that nymph will still have the pathogen, and it will have it through all of its life stages. So that's how the ticks actually get the pathogen, and that's why wildlife hosts play such an important role in Lyme ecology.
Got it. Thank you. Boy, the questions are really good, and they continue to come in. So back to luring ticks in. Mike Heath asks, are there tick pheromone lures available for tick surveillance?
I've seen some studies out there that have looked at effects of pheromones on, sort of attracting ticks. I haven't really heard of any being used commonly. The one that you hear the most about is sometimes using CO2, e.g. dry ice, to sort of lure ticks in. So sometimes you'll hear about folks like who use, the flag like I have here, but then they might put dry ice, either have some contraption that allows it to diffuse out from the pole here, or they'll actually place it if it's a drag. Sometimes they'll even place it on the drag in hopes that that will help attract even more ticks than you're already attracting just by being present. And I have seen that, it does seem like that oftentimes will increase the number of ticks that you collect, but a lot of times it seems to be dominated by Amblyom americanum, the Lone Star ticks, which are probably the most aggressive questers. And a lot of tick biologists will tell you don't need any more of those anyway.
Man, yuck. Questions keep coming. Now we're thinking about fire again. This might be more than you have time to answer, but a question was, what is the correlation between burn times and frequency and tick abundance? What tick life stage should be targeted for maximum control?
Oh yeah, that's a great question. So like in our study, we were working at sites that had a variety of different, burn frequencies, burn intensities, all that kind of stuff. Generally, they were all, for the most part, low intensity burns. But we certainly, we had some sites that were burning every other year. Other sites were burning closer to every 3 to 4 years. And we found in our particular study that there wasn't really any that didn't really seem to play a role. As long as fire was happening on a regular basis, regardless of what the frequency was, we were seeing those sustained reductions in tick populations. As far as what life stage to target, I haven't seen a lot of data out there that's specifically looked at that question of does time, does when you burn affect, efficacy of tick reductions? Because for the most part, land managers aren't burning to reduce ticks. Normally they're burning for other management objectives, and it's just a happy coincidence that it's also knocking ticks back. But with that said, you're mostly going to be burning the larvae and the nymphs for the most part, because most folks are doing dormant season burns, and that's just what happens to be out during that time of year. That, and then of course, black-legged tick adults. But yeah, again, it would be a fascinating research project to look at, if you do a growing season burn versus a dormant season burn, does that, affect efficacy? But yeah.
Yeah, but fire is good.
So. Yes, fire is good.
Yeah. I see we have just a minute till the top of the hour, and we have a few more questions. I recognize that folks are going to need to jump off and take care of other things, but I think Liz is able to stay on for a few more minutes to help answer some more questions. So before everybody jumps off, please join me in a virtual huge thank you to Dr. Gleim for our webinar today. We really appreciate your time, and this is awesome. So thanks pleasure. Everybody. Yeah, thanks everybody for joining us, and we will continue with questions, and we understand if you have to jump off in just a minute here. Okay, We had a brief commercial break. Jenna suggests Insect Shield is a really good resource out of North Carolina. And as I mentioned to you, Liz, last week, I got to see a presentation about a University of Rhode Island study that replicated a North Carolina study on the effectiveness of permethrin, which Insect Shield treats clothes with. So I would echo that from what I saw as well. Okay, back to our regularly scheduled program. Now we're thinking about animals and acaricides. So Jason asked, could climate be why there are hotspots? Cooler temps seem to make them more abundant, at least where I'm from in Massachusetts. Does acaricide affect other animals, such as birds of prey? And someone, Monica echoed, I too was concerned about the effect of pesticide used on rodents and how they affect predators.
That's a great question. And I have not seen any literature on that. It may be out there. I'm just not familiar with it. But I mean, I would certainly second that that's definitely a concern that needs to be further investigated for sure.
Yeah, thanks. Let's see. We also had a question about natural predators that control ticks. What species eat ticks, and is there a correlation between increased tick populations and decreased predator populations? Perhaps bird species that feed on ticks?
Yeah, so I'm sure everyone's heard about Guinea fowl feed on ticks, and other sort of ground feeders. There's actually been very little formal research actually looking at that, but I am aware of, I think, only 1 or 2 studies that have examined whether, for example, guinea fowl could actually reduce tick abundance. And for the most part, from what I gather from those 1 or 2 studies, it looks like they aren't really that effective at reducing tick populations. Another common species you sometimes hear about is possums are really good about grooming ticks off of them. And do they do those things? Yes. Is it really enough to make a dent in the tick population? It seems like probably not. Does that answer the question?
I think so, but Monica also clarified native species would be her primary interest. Are there native species that could be help control tick populations?
Yeah, again, no, not from anything that I've seen.
Okay. So maybe another argument for fire.
Yeah.
So this is a not happy question, but Sean asked, so for employees in the public sector, would it be fair to say that land managers, scientists, et cetera, are statistically quite likely to contract a life-debilitating disease from ticks?
Sorry, Amanda, you kind of you cut up there for a second. Could you repeat that question?
Sure thing. So Sean writes, so for employees in the public sector, would it be fair to say that land managers, scientists, et cetera, are statistically quite likely to contract a life-debilitating disease from ticks?
Oh, that's a great question. Is it a hazard? Yes. And are you more likely to encounter ticks? Probably so, because you're spending more time outside. I will say we actually did a study that we published a year or 2 ago where we were actually looking at tick bites in outdoor workers, essentially forestry and wildlife professionals. And we didn't have a control population, so we couldn't necessarily control to folks that aren't in those professions. We certainly found, yes, you are being bitten by ticks, somewhat regularly. But the one thing that was really encouraging to see was that out of, I think, the 350 ticks or so that were submitted to us, only 3 of them were engorged, which indicated that most of the ticks seemed to be being removed fairly quickly after attachment. And we suspect, again, we didn't have a control population to compare it to, but it seems like, generally speaking, those of us who work outside, you are aware of the risk of ticks. And so you are probably going to be a lot better about protecting yourself and checking yourself than, say, the average person. And again, the best thing you can do to protect yourself is to remove the tick, well, assuming the tick is biting you, remove it promptly. If you can remove it promptly, the chances of it being able to transmit a pathogen, should it be carrying one, is incredibly low. And the good news is it seems like forestry and wildlife professionals seem to be knowledgeable and do that pretty well. So yes, is it a risk? For sure. Do you need to be vigilant? Most definitely. But is it cause for complete panic or avoidance of going outside? No.
All right. That might be, oh, we're getting more questions. Okay. I was going to try to end on a positive note, but we have another really good question that I'm going to not ignore.
Okay.
Adam just asked, are there differences in tick life cycle seasonality between more northern latitudes versus southern, and implications for management treatment to reduce tick populations?
Yeah, so that's a great question. Yeah, so phenology, that is, when different species and life stage are active during the year, is definitely variable by region. And that's one of the reasons it's so important to understand tick phenology in different regions and in different ecosystems. So because there is variation. Now, is that variation going to be dramatic? No, we're talking, maybe ticks are going to become active, a few weeks, maybe a month earlier, say down south than they are up north. You're not going to see, oh, something that's active in the summer here is going to be active in the winter elsewhere. You're not going to see something really dramatic like that. But yeah, you definitely see variation in terms of when ticks are going to be active. And that even changes year to year in the same place because of, weather, whatever, right? So there's a lot of different factors that'll affect that. But in terms of should that affect management, yes and no. If you're taking some sort of action specifically to reduce ticks, then yes, that should change your timing, and it would be valuable for you to know what the phenology is in your particular region. If the management that you're doing, the primary goal is something outside of controlling ticks, then I would say do that at the time that it makes sense to achieve whatever that management goal is. But it's definitely very helpful if you're trying to control ticks, and also, for public health awareness, so that you are aware of when you're at risk. It is really valuable to know what the phenology is in your particular region, for sure.
Got it. Thank you. We had a reminder from Rave Brown to be vigilant, because I think everybody knows somebody, especially people who work in the field, who have been impacted by tick-borne illnesses. So everybody out there, be vigilant. Yes. I think I will try to wind down on a positive note. So Danielle Garneau from SUNY Plattsburgh said, thanks for an informative webinar, and from one tick dragger to another. Thanks.
My pleasure.
All right. So on that note, if you have questions for folks out there, please email Dr. Gleim, egleim@hollins.edu. Thanks everyone so much for joining us, and thank you, Liz, so much for your time and for sharing your wisdom. And we'll post a recording of this webinar on the NASI North Atlantic Fire Science Exchange website, and I'm sure that Catherine's will link to it as well. Thanks everybody for participating, and stay safe out there.
Thanks so much.